Howdy, my name is Jazlyn Gonzalez and I am part of group number 6 with Andres Garza. Today we will be talking about gram, acid fast, capsule, and endospore staining. Our table of contents includes our introduction, gram staining, acid fast staining, capsule staining, endospore staining, and our conclusion. Going on into our introduction. In microbiology, staining techniques are important as they allow scientists to observe and distinguish microorganisms based on structural and chemical characteristics. The gram, acid-fast, capsule, and endospore stains are differential staining methods used to clarify bacteria and visualize specific cellular components that cannot be seen with simple staining. The gram stain is used to differentiate bacteria into gram-positive and gram-negative, based on the composition of their cell walls. Gram-positive bacteria have a thick peptidoglycan layer that allows them to retain the primary stain, crystal violet, even after decolorization. This results in their purple or bluish coloring. In contrast, gram-negative bacteria have a thinner peptidoglycan layer, as well as an outer membrane rich in lipopolysaccharides, which causes them to lose the crystal violet during decolorization, and instead take up the counterstain, safranin, which results in the bacteria looking pinkish or red. The acid-fast stain is primarily used to identify Mycobacterium species, which possesses waxy cell walls containing mycolic acids. These lipids make the cell walls resistant to dyes and decolorizing agents. This technique uses carbofuran as the primary stain, acid alcohol as the decolorizer, and methylene blue as a counterstain. Acid-fast organisms retain the red color of the primary stain, while non-acid-fast bacteria appear blue. The capsule stain highlights the presence of capsules, which are the outer layers composed of polysaccharides or polypeptides surrounding certain bacterial cells. Capsules are non-ionic, meaning they do not readily absorb most dyes. Because of this, a negative staining technique is used, which leaves the capsule looking like a clear halo between the stained background and the stained cell. The endospore stain identifies endospores, which are highly dormant structures formed by certain genera. Endospores form in response to unfavorable environmental conditions and allow bacterial survival in genera, in extreme heat, desiccation, and chemical exposure. The endospore stain uses malachite green as a primary stain and safranin as a counterstain. Under the microscope, endospores appear green within red or pink vegetative cells. The materials that we used throughout the whole lab included the following. For cultures, we used Staphylococcus aureus, Bacillus cereus, Salmonella typhimurium, mixed culture, which included Staphylococcus aureus and Escherichia coli in tryptic soy broth, our unknown number 136, unknown number 276, Mycobacterium sp, and Klebsiella pneumoniae. For the reagents and stains that we used included crystal violet, grams iodine, decolorizer, safranin, quinones carbofuran, acid alcohol decolorizer, methylene blue, 20% copper sulfate, and malachite green. For our equipment, we used a microscope glass slides, bibulous papers, wax pencil, butane tank, deionized water, an inoculating loop, a hot plate, a staining rack and tray, microscope oil, and lens cleansing sheets. Before each stain, we had to do smear preparation. From broth to slide, we had to get one clean slide, use the wax pencil to etch a big circle and label with the culture that we would be using. We then flip the glass slide onto our staining tray so that the etched side is facing down. We swirled the tube of bacteria to ensure that they are in suspension, as they tend to settle to the bottom. We sterilized the inoculating loop, then transferred a drop of the culture broth to the unetched side of the glass slide and spread it to the size of our etched circle on the back side. We then placed our slide on the hot plate to dry. Once dry, we heat fix the slide by passing the slide 3 times through the butane flame. For the smear preparation of a solid culture to the slide, we used one clean glass slide, etched a big circle using a wax pencil and labeled our culture that we would be using. We then flipped the glass slide onto our staining tray so that the edge side is facing down. Using a sterilized inoculating loop, we transferred a drop of deionized water to the center of each glass slide. Then, we sterilized our inoculating loop to transfer the culture onto its designated glass slide, making sure to spread it into the drop of deionized water and smear to the size of our etched circle on the backside of the glass. We then placed our slide on the hot plate to dry. Once dry, we heat fix the slide by passing the slide 3 times through the butane flame. Now for our gram staining. We put the air-dried heat-fixed slides of S. aureus, B. cereus, S. tifamurium, our mixed culture, and our unknowns number 136 and number 276 onto our staining tray, making sure that the bacteria side was facing up. We flood the smears with crystal violet and let it stand for 1 minute. We then rinsed with deionized water for 5 seconds, proceeded to flood with grams iodine mordant and let stand for 2 minutes. We then rinsed with deionized water again for 5 seconds. Lifting the glass slides off the staining tray, we decolorized with the decolorizer and made sure to immediately flush with water for 5 seconds. We then placed the slides back onto our staining tray and proceeded to flood with safranin for 1 minute. We then rinsed with water for 5 seconds and made sure to blot dry with bibulous paper and examined under oil immersion.
For our results and observations, we can see that for our mixed culture, it was gram negative since there is a pinkish red color. However, no other observable markers were seen. This might have been due to human error with the microscope or just improper staining. As for S. aureus, it was gram positive since there is a purple color to it, meaning that there is a thick peptidoglycan wall. We were also able to identify that there has been clusters of spherical cells, which is known as cocci. As for B. cereus, this was also a gram positive staining, meaning that there was a thick peptidoglycan wall, and we were also able to identify large rod-shaped cells known as bacilli in a chain-like formation. Lastly, as for S. tifamurium, this was a gram negative stain, which meant that it had a thin peptidoglycan wall, hence the pinkish red color, and the cells were also short rods. For the results and observations of our unknowns, for unknown 136, this was a gram-positive staining, meaning that it had a thick peptidoglycan wall, and we were also able to identify that the cells were spherical, meaning that they were identified as cocci. As for unknown 276, this was gram-negative, hence the pinkish red color to it, meaning that there is a thin peptidoglycan wall.
Now for our acid-fast staining.
For our results and observations with the acid-fast stainings, we can see on the left-hand side, the Mycobacterium sp appeared to have slender rods and clustered, and they also appeared to be blue even after the methylene blue counterstain. As for the acid-fast stain on the right, the Mycobacterium appeared with a red staining, meaning that there was retention of carbofusion due to the waxy mycolic acids.
Now for our capsule staining. Using 3 clean glass slides, we placed a drop of crystal violet onto one. Using aseptic techniques, we added a loofah of cane ammonia to the slide, mixing it into the dye, but made sure not to spread it out. We then used the second slide to drag the stain cell mixture into a thin film along the first slide and air dried for 5 to 7 minutes without fixing it. On the third slide, we placed a drop of deionized water, followed by a loofah of cane ammonia. Using the clean glass slide, we spread out the bacteria similar to the first slide that we did with crystal violet. We then let it air dry until completely dry. We followed it by staining with crystal violet for 1 minute. We then rinse the slide using 20% copper sulfate, not deionized water. We then blot using bibulous paper and found the bacteria on both slides using the microscope and viewed under oil immersion.
As for the results and observations of our capsule staining, we can see that in K. pneumoniae in deionized water, these cells look lighter and the background is faint. So that means that the halos are less distinct than in the crystal violet slide. And although contrast is low, subtle clear zones are still visible. The capsule was also positive with the reduced visibility. As for K. pneumoniae in crystal violet, there were clear, smooth halos which were visible against stained background. The cells do not seem to be stained, however, this could be due to insufficient microscope magnification, and there was a clear posit capsule stain nonetheless.
Now for our endospore staining. We prepared an air-dried heat-fixed slide of our gram-negative, which in our case was our unknown number 276, and placed it onto our staining tray, making sure the bacteria side was up. We trimmed filter paper to fit the width of the glass slide and covered the smear with it. We saturated the filter paper with malachite green stain solution. Made sure to not overflood. We then held the glass slide over boiling water for 7 minutes to avoid drying out, and malachite green to filter paper. Do not overflood. We then remove the slide, let it cool, remove the filter paper, and rinse with DI water for 30 seconds. A counterstain with safranin for 1 minute. We then rinse with deionized water for 5 seconds, and we blot dry with bibulous paper and examined under oil immersion.
As for the results and observations with our endospore staining, we can see that for unknown 136, there was no appearance of green within the endospore staining, and this was most likely due to possible human error during the steam fixing, which did not properly allow for the malachite green to penetrate the spore's resistant coat. Now moving on to our conclusion. So in conclusion, in this lab, we applied 4 differential staining techniques: gram, acid-fast, capsule, and endospore staining to visualize key bacterial structures and classify microorganisms. Each stain revealed unique information. Gram staining distinguished bacteria based on cell wall thickness. Acid-fast staining highlighted the waxy mycolic acids of Mycobacterium. Capsule staining showed the protective halos that increase virulence of K. pneumoniae, and endospore staining revealed the survival structures of Bacillus subtilis. Our observations confirmed textbook expectations. Gram-positive bacteria such as Staphylococcus aureus and Bacillus aureus appeared purple, while gram-negative bacteria like our mixed culture and Salmonella typhimurium appeared pink. Acid-fast Mycobacterium (SP) retained a red dye despite decolorization. Capsules appeared as clear halos against purple cells and endospores, and for our endospores, they failed to stain green within the red vegetative cells. The unknown samples could also be partially classified, showing how differential staining is a practical tool for microbial identification. Overall, this lab demonstrated that staining is not just about color, it's about fundamental diagnostic methods that connect bacterial structure to function. By practicing these techniques, we gained a deeper understanding of how cell wall composition, protective coatings, and survival mechanisms influence microbial behavior, virulence, and clinical significance. These skills are essential for microbiologists and healthcare professionals as they provide the steps in accurately identifying pathogens and guiding treatment decisions. These are also our resources that we used for our lab. And thank you guys for listening to our lab report video.